Welcome to my channel. In today's videos, I will be talking about type 2 hypersensitivity reaction, also called as cytolytic reaction. Cytolytic means cellular breakdown. It means the destruction or breakdown of cells is going to be happen in this reaction. Okay. Examples which include heparin-induced thrombocytopenia, autoimmune hemolytic anemia, blood group mismatch reaction. All the detail and the mechanism of these diseases, these reactions, it will be discussed in the later part of the video to make your concept very clear. So first of all, let me tell you one thing. What will happen in this case? In this case, for example, this is a tissue. In this case, antigens, foreign antigens or self antigen, they might come and attach on some cells of the tissues, some cells of the tissue. Foreign antigen or some auto antigen, self antigen, they can come and attach on the tissue. But let me tell you one thing, they must have to fix, they must have to attach, they must have to adhere on the cell surface. Okay, they must have, and once they are attached on the cell surface, the antibody will come against these antigen and it will attach over them. And once this antibody has been attached, you know what, this antigen, in the case of type 2 hypersensitivity, this antigen could be IgG or it could be IgM. And once this antibody has attached over these antigen, these antibodies can cause the cell lysis, can cause the breakdown of cell. How? There are basically two mechanisms of cellular destruction by antibody. The one is complement dependent and other is complement independent. The detail of both the mechanism will be discussed in the later part of the video. Okay. So once the cell has been destroyed, ultimately the damage will be caused to the whole tissue. So we can say that antibody mediated cellular lysis occur in type 2 hypersensitivity reaction, which can result in the tissue damage. So it means tissue damage will be caused by cellular lysis, and this cellular lysis will be antibody mediated, and the antibodies are IgG, or they could be IgG or they could be IgM. Okay. Now, if we talk about some examples, heparin induced thrombocytopenia. Heparin is an endogenous molecule. Okay, so once it will enter in our body and it will adhere to the, it will attach to the platelet surface. Platelet surface. Against these antigen, antibodies will be produced by our B cells, and that are IgG and IgM antibody. They will come and they will attach on these heparin or antigen that is present on the platelets surface. Now what will happen? We know that this is the structure of antibody. This portion is called as Fab and this portion is called as Fc region. The Fab portion is for the attachment with the antigen. Here you can see the Fab portion is for the attachment with antigen, while this Fc portion can activate complement system. Complement system. Like for example, this complement, there are several complement proteins present in our body, like C1, C2, C3, C4, C5, all these proteins, they are present in our body. But what happened? This antibody, it can cause activation of that complement protein once there is antigen-antibody interaction. And once this antigen-antibody interaction has been taken place, this Fc portion of this antibody, it can cause the C1 protein activation, and once this C1 protein has been activated, it will attach with this Fc portion of the antibody. Now this C1 has been activated and attached with the Fc portion. Now this C1, it will be able to attach C2b. Actually this Fc portion will convert C complementary protein into C2 complementary protein into C2A and C2B. The C2B will attach with this C1. The detail of the complement system has been given in my previous video. Now this C2B, they will convert C4 into C4A and C4B. The C4B will attach with this complex, and C4 it will stay away and perform some other function. Detail has been discussed in my other videos that are related to the complement system. Now this C1B, C2B, C4B, they can convert C3 into C3A and C3B. Now C3B will attach with this complex, and C3A it will stay away. Now this whole complex, they can convert C5 into C5A and C5B. Now C5B can attach C6, C7, C8 and C9 protein along with this, and this complex of C5B, C6, 7, 8 and 9 is called as MAC, membrane attack complex. These proteins, they all will get themselves arranged to form this pore-like structure. They all will arrange themselves to form this a pore-like structure. Now this pore-like structure will be placed on the surface, on the cell membrane of the platelet. Once it has been placed on the surface of the platelet, this complex has been placed on the surface of platelets, what will happen? The water start moving inside the cell. So because of the osmotic pressure, the cell will burst, it will burst, it will be broken down. Similarly, all the essential components, they will start leaking out of the cell, and ultimately the survival of cell will not be possible. In this way, heparin induced thrombocytopenia will happen because of type 2 hypersensitivity reaction. Because IgG antibody or IgM antibodies are involved, complement system is involved, and cellular destruction is being taken place. So one more thing, here you can see the complement system is involved in the breakdown of the platelets. So what do you think? It will be complement dependent cellular lysis or complement independent? Obviously, this type of cellular lysis will be called as complement dependent. It's a complement dependent cellular lysis. Similarly, autoimmune hemolytic anemia, what happened on the RBCs? Some antigens, some self antigens are present, but in this disease, this is an autoimmune disease. So if we know that in the autoimmune disease, our B lymphocytes, our T lymphocytes, they start producing antibodies and they start producing damage to our self antigen. So in this case, these are the self antigen that are present on the red blood cells, but this is a disease condition. So what happened in this case? Auto antibodies against these antibodies start producing, and they will start binding on the surface of the red blood cells. Okay, now what will happen? The complement system will activate and it will cause the cellular destruction in this pathway. It will cause the MAC will be formed, MAC will be placed over there, and cellular destruction will be taken place again by the complement dependent cellular destruction. Similarly, blood group mismatch reaction. For example, a patient have blood group A. So it means its red blood cell will be having A antigen on the surface. Its RBC will be having A antigen on its surface, while his blood will be having anti-B antibodies. Anti-B antibodies as well. Okay. Similarly, for example, by a mistake, a patient having blood group B donate its blood to this patient. Now what will happen? For example, now this B blood group has entered in its surface, having B antigen on the surface of red blood cells. But we know that the recipient was having anti-B antibodies. Now these anti-B antibodies, they will attach on these B antigens, and they will start cellular damage by complement dependent pathway by producing MAC protein. Okay, in this way, the blood group mismatch reaction is also the example of type 2 hypersensitivity reaction, in which cellular destruction, cellular lysis is going to be happen because of antibody. So we can say that antibody mediated cellular lysis. Well, there is another example, that is complement independent, in which complement system is not involved. For example, this is a healthy tissue, nucleated cell, platelets, they are non-nucleated cells, they are just fragments. RBCs, they are non-nucleated cells, they do not have any nucleus. But on the nucleated cell, we know that this MAC will not be able to form. This point is very important, listen to me. For example, this is a nucleated cell, antigen has been attached on the surface of these cells, and antibody has been formed, and antigen-antibody interaction has been taken place. But MAC will not be formed. If MAC will not be formed, obviously the cellular destruction that was being taken place with the MAC, it will not take place. Okay, now why MAC is not being formed? Because complement regulatory proteins are present on the surface of nucleated cells. Complement regulatory protein, or we can say that membrane bound complement regulatory protein, membrane bound complementary regulatory protein. And these protein, what they do, they stop the conversion of C3 into C3A and C3B. They stop the conversion of C5 into C5A and C5B. They stop the conversion of C4 into C4A and C4B. So once all these conversions will be stopped, do you think this membrane attack complex will be formed? Obviously, this membrane attack complex will not be formed. And once this membrane attack complex will not be formed, how these antibody can destroy these nucleated cell? Because complement system is not involved. So this type of destruction will be called as complement independent. Now let's see how complement independent pathway works. Now what will happen? MAC will not be formed obviously, but our natural killer cells, they are type of lymphocytes. Lymphocytes can be T lymphocyte, they can be B lymphocyte, they can be natural killer cells. So they are the lymphocytes. They do have a receptor for this Fc portion of IgG or IgM antibody. So once these antibody has attached on the nucleated cell, the natural killer cell, they will come and it will be having receptor for the Fc portion of these antibody. So the interaction between the Fc portion and the natural killer cell will be taken place. And once this interaction has been taken place, the natural killer cell, it will release two very important enzymes. The one is perforin, another is garanzyme B. Perforin and garanzyme B. Now, perforin is a small pore-like protein. It will cause small pores in the nucleated cell, and from these pores, the garanzyme B will enter inside the cell. And once garanzyme B has been entered inside the cell, it will cause the apoptosis of the cell. How? It will basically activate the caspases protein. And you know that caspases are those proteins which can cause the lysis or breakdown of the cytoskeleton proteins of cytoplasm or cytoskeleton proteins of the nucleus. So automatically apoptosis will be taken place. So the cellular destruction will be taken place. So in this pathway, you can say natural killer cells are involved, but complement system is not involved. So this type of pathway, this type of damage will be called as complement independent pathway. So this was all about the type 2 hypersensitivity reaction, and different types of example I have discussed, and different types of pathway and their mechanism has been discussed. I hope you like my video. Please don't forget to subscribe my channel and press the bell icon so that you may get notification for the future videos in which I will be explaining about type 3 hypersensitivity reaction and type 4 hypersensitivity reaction. And like and share this video as well. Thank you so much.